Hi, everyone. My name is Beth Sheets, and I'm a program manager at UC Santa Cruz Genomics Institute. In this session, I'm going to introduce you to GWAS resources in BioData Catalyst. First, I want to state I have no financial relationships with relevant companies to disclose. In this session, I'll give an overview of GWAS resources available across the entire BioData Catalyst ecosystem. Then I'll break down one example user path that leverages PIC-SURE, Gen3, Terra, and Dockstore, which are components within the ecosystem. And then I'll give a live demo of a GWAS in the Terra workspace. In the next session, you'll be able to see a GWAS example in 7 Bridges. Phenotype-genotype association tests can help us identify genetic risk factors for heart, lung, blood, and sleep disorders. We know the majority of variants identified through whole genome sequencing are rare, and BioData Catalyst offers access to hundreds of thousands of whole genome sequences that offer the statistical power necessary for discovery. Beyond accessing the rich repository of cloud-hosted data, BioData Catalyst can also help you find data of interest and learn how to apply for access to controlled access studies. Bring your own data into the ecosystem to compare to our hosted data, collaborate in secure cloud workspaces. We also have a rich repository of available analyses that you can reuse or bring in your own. This includes interactive analyses, for example, in Jupyter Notebooks or RStudio sessions, and also scalable analyses that you can leverage through workflows. We also have several hosted applications for helping you analyze your data and features to help you monitor cloud costs. One example of hosted data set I wanted to point out is the TopMed program, which provides rich phenotypic and genomic resources. Currently, there are 44 high priority clinical and demographic variables that have been harmonized previously by the TopMed Data Coordinating Center that you can leverage in your association tests. And you'll also have access to the non-harmonized raw, which is the raw clinical and phenotypic variables that are available for hosted studies. BioData Catalyst currently hosts Freeze 5B and 8 of the TopMed program, where Freeze 8 includes 158,000 whole genome sequences. This means that you'll have access to the CRAM files and their associated CRY files, jointly called BCFs, that have been subsetted by study and consent group. If you're part of the TopMed consortium, you can access the combined exchange area that hosts GDS formats of the variant files, kinship files, annotation files, etc. On the right, you can see that the TopMed program is quite diverse, with a breakdown of ancestry and ethnicity in this first pie chart. And then the bottom, you can see the phenotypic focus of these studies. When you use a BioData Catalyst workspace, you also have the potential to leverage data that's hosted in other NIH cloud ecosystems if you have access to those data sets through their data access mechanisms. These other NIH cloud ecosystems have rich multi-omic and trait data available, and you can discover data that is currently hosted in the cloud using the NCPI data portal, which is linked in this slide deck that you'll be able to access later. For example, for GWAS researchers, this means that you might be able to easily access GTEx data that's hosted by NHGRI Anvil, and you can access this using a GA4GH data access standard, which is cloud agnostic and available to researchers in both BioData Catalyst workspaces. On the bottom, you can see that the Gen3 service, which is also available in BioData Catalyst, is used by the Anvil ecosystem. And you can see how we've filtered down our selection on the GTEx dataset. And you can hand this off into cloud workspaces. You can also bring your own data where you can upload data into your cloud workspace. but we want to remind you that you should only do this if you have the appropriate approval and that you're not violating the terms of your data use agreements, limitations, or IRB policies and guidelines. On our website, we have some examples of how you could add a cloud use statement to these agreements. Data that you might be interested in bringing in for GWAS include things like harmonized trait data that's been developed from a working group that you're a part of, annotation files, or potentially new types of variant files like structural variant calls. You can learn more about how to bring your own data in the linked document on this slide. We have several services that can help you find data of interest and build cohorts. One of those is PIC-SURE. You can explore data and find projects to apply for access with what's called open PIC-SURE, where you don't need access to any prior data. You can explore our data assets in an open access way. This is available as both a easy to use user interface, or you can call the PIC-SURE API in an R or Python Jupyter notebook within either cloud workspace. Using PIC-SURE, you can search across hosted data using queries to build cohorts, where you can explore already harmonized variables or discover new traits to be harmonized across target studies. Another service that helps you explore our data assets is Gen3. Gen3 has indexed the data that is hosted in the cloud by NHLBI and organized it into a data model for easy cross-project analysis. They offer a faceted search feature where you can explore by project ID, harmonized variable, or file type. And one example use case is that you can select projects for which you have access and hand those off to Terra or Seven Bridges workspace. Once you have your data access and you're ready to analyze the data, you can do that in a collaborative and secure cloud workspace. There are two options in BioData Catalyst. One is Seven Bridges that you learned about earlier today, and the other is Terra. Both of these workspaces offer features so that you can control who can access your workspace, or you can work in a private workspace. This is the place where you would be able to bring your own data and host it in the ecosystem. You can organize your data, methods, and results with various features. You can also share the data that you uploaded across workspace with appropriate collaborators. You can launch interactive analysis sessions and also scalable workflows. And there are features in workspaces to help you track costs associated with the storage and compute of your data. I wanted to give a brief introduction to some concepts that are important when migrating into the cloud. The first is Docker. Docker is a technology that containerizes and reproduces an exact compute environment with all software libraries and dependencies. So this means you spend less time trying to install the appropriate software and dependencies for your analysis. Docker can be used locally on your own machine, but it's pretty necessary in a cloud environment where you request resources for a short amount of time. All of our analyses leverage Docker, including Docker-based workflow languages. And if you've never heard of a workflow language, this is just a way that you're describing your computational analysis. So you can do this and make your analysis portable across compute environments and also different users. So you'll basically define complex analysis tasks that have been potentially written in R, Python, or Bash, and you can chain those tasks into workflows and parallelize their execution. So this basically means that we can describe how to take the output from one piece of software and use it as input into another piece of software. In BioData Catalyst, we leverage two workflow languages. The Common Workflow Language, or CWL, is used in Seven Bridges, and the Workflow Description Language, or WDL, is used in Terra. Reproducible interactive analyses also leverage Docker, and these are used in our workspace environments. So when you leverage Docker, you and your colleagues can run the same analysis in the exact same compute environment. An example session costs about 6 cents an hour. And interactive analyses are used for Python or Jupyter notebooks. If you're in the Terra workspace, you can actually configure your notebook to leverage a Spark cluster for parallel analysis. You can use our studio sessions, our Shiny apps. Recently, via the Anvil Cloud ecosystem, the Terra workspace can use Galaxy. In 7 Bridges, you can use SAS. Some example interactive sessions that might be of interest to GWAS researchers are kind of showcased here. These are available in the Terra workspace. There's notebooks to help you prepare your files for input into workflows. And this includes something like if you would like to merge these VCFs that were originally jointly called but subsetted by project and consent code back together to form your cohort, you'll need to merge and potentially subsample those. And there's a notebook to help you set up your data for use in those workflows. There's notebooks to help you explore the traits of interest for your analysis and select potential covariates for your association test. You can see an example on the right of this. There's also several available notebooks that leverage Hail, which is a Python package that can be backed by a Spark cluster. And in these interactive sessions, you can explore the genomic data that you're interested in using in an association test and account for ancestry and relatedness. You can also use notebooks to explore sample and variant quality. So we talked about different reasons that you might want to use an interactive session when you're more in the data discovery mode. But when you're ready to scale up your compute, you'll need to use workflows. And we have a rich repository of workflows available in Dockstore. Dockstore is another service in the ecosystem, which can be considered an app store for computational tools. Dockstore entries really leverage reusability and portability. And so a Dockstore entry includes things like the link to the source code if it was hosted in GitHub to leverage version control, links to the Docker images that are used in the workflows. There can be example test data available, example parameter files that help you set up a workflow run, extensive documentation, and more. There's features that can help you find vetted tools and workflows that have been shared by the community. This includes tools and workflows that have been verified by the Dockstore team. There's an organizations feature where organizations that you might be familiar with have shared work that they've published on Dockstore. There's a feature where a workflow or an organization can be starred by a user, and that means that that user might like that workflow or organization. And users can also identify themselves by linking their Dockstore username with their ORCID, which would then link you to their scientific body of work. You can launch a Dockstore entry directly into the BioData Catalyst ecosystem with this launch with button. And you can also leverage the Dockstore command line interface to help you develop and test workflows locally before launching it in a cloud environment. Some GWAS workflows that are currently available are showcased in the BioData Catalyst organization on Dockstore. This includes an analysis pipeline developed by the University of Washington Genome Analysis Center that leverages the TopMed pipeline and Genesis R packages. This includes workflows for ancestry, genetic relatedness, and association testing. And I've linked where the CWLs and WDLs are hosted, both in Dockstore, and CWLs are also available in 7 Bridges. There's another pipeline that was developed by a group called Analysis Commons that also leverages the Genesis R package, and those WDLs are also available in Dockstore and linked in this slide. You can see the analysis commons example on the right here. This is a collection within the BioData Catalyst organization on Dockstore. And you can see there in the documentation, there's a little bit more that just information about these workflows, links to tutorials, and also some cloud cost examples when using these workflows. If you don't find what you need on Dockstore, or maybe you have your own analysis that you're developing, you can also bring your own tools into the ecosystem. We have documentation that can help you learn how to port your pre-existing analysis into the cloud by learning about Docker, different secure software packages, and how to write work in these two workflow languages. So this will really make your R, Python, and Bash scripts more interoperable and reusable. You can then learn how to share your work with the community on Dockstore, which is open access and provides a lot of features for helping you publish your computational methods, including setting your workflows up with DOIs for publication and citation. And we hope you'll get started with our Bring Your Own Tools documentation. There's several hosted applications in our ecosystem that you can leverage for GWAS. This includes the TopMed imputation server, which yields phased and imputed genotypes based on the multi-ethnic reference panel available from TopMed. You can use the 7 Bridges Annotation Explorer to explore different variants of interest. There's also the Integrated Genomics Viewer in Terra, which can let you explore tracks from BAM and CRAM files. So I know when you're thinking about transitioning to the cloud, one of the things that's a barrier is really understanding cloud costs. So I wanted to just introduce you to things to think about when maybe you see example benchmarking of workflows that have some associated costs and how that might relate to your own specific use case. Things that it could influence cloud costs for GWAS studies include number of samples you're using and their genetic structure, the number of variants, the algorithm used in your analysis, be it single or aggregate, and if it's aggregate, the number of variants in that aggregation, the implementation you choose, and also the cloud hardware resources that you request when you launch a workflow. For example, CPUs are kind of the most expensive cloud resource that you request, but also you want to think about the memory you request, the attached disk, and whether you choose to use preemptibles. Preemptibles are a way to request resources for a reduced cost, but they're really only effective for short jobs. Another thing to think about is how does this analysis potentially scale computationally? Some things that scale linearly are a little easier to try to plot out how much you expect it to cost as you add more samples. Linear analyses might be something like a file conversion, but many of our analyses are actually nonlinear, so it can be a little bit more complex to estimate their cost. This includes things like the null model and association tests you run, where I've heard that association tests may scale more like quadratically. Some other things to think about are the types of things that you'll pay for. I wanted to start off by kind of emphasizing the things you don't have to pay for when you're using BioData Catalyst. And that includes any data that's actually hosted by the ecosystem. You don't have to copy it and host it in your own workspace. You can just stream or localize that data directly into your compute resource for analysis. And there's ways that BioData Catalyst is set up to authenticate you to access that data. You shouldn't have to pay for any data egress fees. Data is hosted in both Amazon and Google Cloud, and you should do your compute in the cloud where the data is hosted. And there are no markup costs for AWS or GCP resources when using BioData Catalyst. Things that you might have to pay for in terms of storage costs are any data you upload or copy into your workspace storage bucket, any derived data that's created when you run, for example, an interactive notebook or a workflow, the outputs of that will be copied into your bucket that's associated with your workspace. And there's some small fees for any data download. Some compute costs that you might want to think about are the interactive notebooks and RStudio sessions you run. These can be pretty cheap, where a basic session is about 5 cents an hour, but they can get more expensive if you're trying to run more computationally heavy analyses in these environments. For workflows, you might want to think about how many times you'll need to run a workflow in your analysis. Things that you might need to run only once, or something simple like a file conversion. Things you might need to run a few times are in association testing are things like relatedness analyses like LD pruning or PCA or kinship analyses. And things that you're going to need to run many times in your association testing. So again, when you're plotting out a proposal of how much you think this will cost, these are maybe also some things to think about as you write up your budget. Okay, so that ends my overview of GWAS resources. And now I wanted to just give a single example user path. This isn't the only path, but I wanted to emphasize how all the services in our ecosystem work together and build this really rich resource for you. But it's sometimes complicated to understand how they all fit together. So here's one example of how I might leverage different parts of the BioData Catalyst ecosystem. I don't have access to any data assets in BioData Catalyst first, so what I want to do is find some data to apply for access, and I can do that using Open Picture. All I'll need to log in is an ERA Commons ID. Once I find some projects that I'm interested in, I will request access to those using potentially a dbGaP proposal. And once I'm granted access, then I'll need to pull together some cloud costs. There are free cloud costs available from NHLBI to new BioData Catalyst users, so I can access those on our landing page. And once I have data and cloud costs, I'll need to pick a workspace environment that I want to work in. In this example, I chose Terra. And in my Terra workspace, I need to set up things like linking my cloud credits. Once my Terra workspace is ready to go, I will log into Gen3 and find the data for which I have access, select the cohorts that I want to use for a particular study, and click handoff to my Terra workspace. In Terra, this is where I can invite my named collaborators. I can start to organize my data into data tables and explore my data with both interactive notebooks and also launch scalable workflows in WDL, the WDL language. So BioData Catalyst doesn't host all of the data that I want for my particular analysis. So I'll have to bring my own data, like some harmonized variables developed by the Top Med working group that I'm a part of. But simultaneously, I might also just want to see how others have run a GWAS before in Terra. So I'll explore a tutorial that is hosted as a featured workspace in Terra. And here I can get some example code that has been used before to explore traits and genetic relatedness, and also see how people have set up their data for running in association tests. So I can reuse those notebooks by copying them into my own Terra workspace and editing them for my use case. I know that I also want to do some more scalable analysis, including an actual association test. So I can go into Dockstore and search what available WDL workflows that they have for GWAS. And I decide that I need workflows for VCF merging and subsampling, because I want to merge some VCFs that were subsetted by consent code back together. I need to account for relatedness in the genetic samples, And I also want to port over association tests from the Analysis Commons collection. So I can launch each of these workflows directly into my Terra workspace and conduct an association test. But I also want to do some follow-up analysis on some of the outputs of my association tests. So I want to actually bring my own tool into BioData Catalyst, and I can leverage the documentation on that, where I can create and update a Docker container and wrap my Bash or Python script that I've been using locally in the WDL language, and then launch it from Dockstore into my Terra workspace. So I hope you can see how all of these services can work together to build a lot of really amazing features that I can use for a GWAS study. Next, I wanted to just give a brief demo of a GWAS that leverages Gen 3 and Dockstore and uses the Terra workspace. So I already have access to data, and I've already set up my Terra workspace, or my Terra account, and added cloud costs. So I want to go in and find the data that I'd like to explore in my GWAS. So I'll click Learn. Oh, no, I won't. I'll click Services, and I'll go to BioData Catalyst powered by Gen 3. And I'll launch Gen 3. I'm already logged in. I can log in with my ERA Commons, but I'm actually also logged in as a developer here. Once I'm logged in, I can hit Exploration. I'll hit Data with Access. Because I'm a developer, I have access to all of the projects. But for today, I want to use a tutorial data set to get familiar with how a GWAS might be run in Terra. So I'll hit Tutorial, and I'll select the tutorial synthetic data set that is pointed out in the Terra GWAS tutorial. Now I'll export all of this data to Terra. So I'm right now just exporting one project, but if I wanted to build a cohort of multiple projects, I could select them all here and hand off that entire cohort. You can see down here that my export is in progress. This shouldn't take too long. And I want to emphasize when I'm exporting data from Gen 3 into a cloud workspace, I'm not actually copying the large genomic files over. I'm copying links to those genomic files. So in this way, I'm not paying to host any of this large data. I'm only copying over basically text to data tables in my Terra workspace. So I can point out interoperable space here where we were in Gen 3. And now that I've handed off data, I'm moving into Terra. I have a few options here for where I'd like to put this data in different workspaces in Terra. If you're running this later, I would suggest you start with the template and import directly into the BioData Catalyst GWAS 1000 genomes tutorial. I did that before, and because I already reran everything for the sake of time, I'm gonna just go into the workspace where I already ran this. So my July copy. And now I have a prompt that my data import is in progress. However, because I ran this already before, you can see that the data has populated into several data tables in my workspace. These data tables are organized into and relate in a graph that was developed by Gen 3. And this graph structured data model is useful for comparing data across many different projects in Topman. You can learn a little bit more about it in our documentation. In the workspace environment, I want to point out a few things. First, there's a dashboard, and this is basically documentation about the workspace. You can keep notes here, or if you're using a featured workspace like this GWAS tutorial, it'll give you an overview of how to run through the tutorial. So the first thing that I'm going to do in this tutorial is explore the data using some interactive sessions to get familiar with the data and make specific decisions. The first notebook available just shows the user how to prepare Gen 3 data for exploration. So it's in this kind of graph structure where the tables relate to one another, and there's example code that helps me pull this into a single consolidated data table using the data that I'm interested in. You can see an example of that here. This is something that you might be familiar with already. It's kind of like an Excel sheet that we've basically uploaded into Terra. You can also bring your own Excel sheets formatted in as a TSV file and upload them into Terra as well, if you prefer that route. And so this has our samples and different trait and demographic information. The next notebook that I want to show you is, I'm just going to do a preview, is a notebook where I can kind of get a better understanding of the trait data and look at visualize different distributions in my data and make some decisions and select which cohort or covariates I might want to include in my association test. So you can see we've provided some example code for visualizing different kinds of plots. And I can input in a variable of interest. For example, here I did LDL. At the end of this notebook, I'll make a CSV file of the trait data that I'm interested in using in my association test. The next notebook I wanted to show is one that helps me do a little bit of genomic data preparation. So I might have a new cohort and I want to see or explore ancestry and genetic relatedness in that cohort. I know I need to account for genetic structure in my association test because it can confound my test. So this is an interactive way to look at your genomic data. You can also do ancestry and the relatedness analysis using workflows, but this one is a nice teaching tutorial so you can learn what you're trying to think about and account for when preparing your data for association testing. This leverages the Hail Python package, which can also use Spark clusters. I can configure my interactive cloud environment session to use Spark, which basically allows me to parallelize some of the processes that I might run using the Hail package. So I'll just briefly scroll down and you can see some visualizations that I might want to look at when I'm exploring my data. Like I might want to get more familiar with what types of information is available in the VCF that was provided. And I might subset some things, for example, filtering my minor allele frequency. There's code in here for LD pruning, for running PCAs and visualizing PCAs and seeing if I need to account for genetic structure, generating genetic relatedness matrices, and so on. At the end of this notebook is another handy snippet of code that you could steal. And it's basically taking all of these files that I've generated for my association test and organizing them into a new data table called sample set, where I can keep a record of all of the data I've used as inputs into my workflow. And I can also actually add outputs there as well. So I'll show you what that looks like when I go back out to my workspace and go to data table. I'll go to sample set. And I can see that I have one example that I've set up here. I can name this something useful to me as a record of what I tested in this specific run through and the decisions that I made, the covariates that I selected, and so on. And I can actually use this data table in sync with how I'll launch a workflow to kind of keep a good record of the workflows that I'm running. So I'll move over to workflows. And you can see in this tutorial, we've already imported in 2 workflows available from Dockstore. I can just right click and you can see what Dockstore looks like and how it's integrated into the workspace environment. Dockstore entry has several different features. You can look at the source code on GitHub to learn a little bit more. There might be some more example documentation there. And you can see how you could easily launch this workflow into BioData Catalyst. However, it's already hosted in this tutorial workspace. So I can show you quickly how we leveraged the data table feature with some of the workflows. So this is the workflow that's actually going to run a null model, an association test, and output a QQ and Manhattan plot. And for the inputs, I'm going to run a workflow with inputs defined by the data table that I showed you called sample set. And I can select, we only have one example here. And basically I'm going to pull data for this row that I've selected from specific columns as inputs into my data table. So for example, the genotype files that I want to use from this selected row of this sample set, I'll use what's listed in the GDS column. So it'll have an array of GDS files that represent each chromosome of the cohort that I'm looking at. For outputs, I can also append every output from this workflow, which there are several, back to that same sample set data table. Then everything is kind of organized, and I can see for a specific set of parameters that I chose, what was the output from that parameter. So it's a nice way to organize my methods and results. So once everything is set up, I can hit run analysis. This will launch the job in Google Cloud, and Whittle workflows actually leverage a specific execution engine called Cromwell. And so Terra on the back end does all of this for you. So all I have to do is set up my inputs, and it will send off the workflow for compute. In the job history, I can monitor how this workflow is going, including looking at some logs to see how it's doing, and monitor how much time different steps are taking. For example, you can see I've run this just a couple times. You can see that this prior example that I ran through succeeded. And I can just briefly show you from the job manager page how I can get a little bit more information about this workflow that I ran, including inputs and outputs, and the execution directory on Google Cloud that can give me things like log files. And I can briefly show you that the outputs of this include some different CSV files of the data, and also a nice little summary plot, so I can see just briefly how my association test went. This is a ping file. It's linking me to the Google bucket. So Terra will take the files from my analysis run and copy them into the Google storage bucket associated with my workspace for long-term hosting of the data. And here you can just see an example QQ plot and a Manhattan plot. This was using downsampled data, so there's just a few variants here, so it kind of looks like a different Manhattan plot than normal. But yeah, that ends my demo. If we go back to the slides, you can see that I've attached some screenshots here if you're interested in trying this out on your own. And I just wanted to acknowledge NHLBI and our BioData Catalyst consortium members for all their support in creating these GWAS resources. And I wanted to give a special shout out to Elisa Manning, Tim Majarian, and Stephanie Gogarden. And I also just wanted to say, if you are interested in using the BioData Catalyst ecosystem later today, Amber will give an overview of how you can join our ecosystem and leverage some of these resources. And with that, I will take questions. Thanks for your time.